Good afternoon, everyone. So what I'm going to do today is introduce the field of genetic epidemiology and talk about the research methods that we use in this field. You'll see that because our bread and butter in this field of genetic epidemiology is to unravel the genetic contributions to complex disease, and last week you heard about complex disease, there will be some overlap with the concepts that Dr. Leibel introduced last week. But we've been talking while I've been preparing my presentation over the week, and we agree that the concepts that overlap bear repeating. So here we go. Not sure exactly where to point, but on the screen. Okay. So genetic epidemiology is a fairly young discipline. The term was first coined in 1978 in a book by Morton and Chung. And it's a field that's aimed at the study of the genetic contributions to the causes of complex disorders and interactions between genetic and non-genetic factors in disease etiology. And I like this definition from D.C. Rao, who is one of the fathers of this field, because he points out why we really needed a new field. He says that it's an emerging field that represents an interaction between the two parent disciplines, genetics and epidemiology, but it's different from epidemiology because it focuses on genetics and family resemblance. It also differs from population genetics because it focuses on disease. And traditionally, population genetics was really focused more on basic questions about evolution and not so much on disease. And it also differs from medical genetics because it's talking about population aspects. And medical genetics, like all fields of medicine really, is focused on individual patients. So we needed a new synthesis, and this really grew up it began in the mid-1970s. So we focus in this field on complex disorders, and these are disorders that are common, and they are really all of the disorders of major public health impact. And we call them complex in part because their genetic contributions are inconsistent with straightforward Mendelian inheritance. And what are the complexities? Well, the effects of multiple genes, each of which has a small or moderate effect on disease risk. And they can be polygenic or oligogenic. Polygenic is the word we use when we're talking about a very large number of genes contributing to the phenotype. And oligogenic is the term we use when we're talking about a smaller set of genes. Complex disorders involve gene-gene interaction or epistasis. They also involve gene-environment interaction. And another important concept is that they involve de novo genetic variation. And over the past decade or so, and really very much in the last few years, we're beginning to recognize the importance of de novo genetic effects. And these are non-inherited genetic influences on disease, so that they occur during the process of gametogenesis, leading to a patient who then develops the disease. So we're really starting to recognize that this is very important, and it's a non-inherited form of genetic variation that influences disease risk. So one of the most important concepts in this field is the difference between genotype and phenotype. And the genotype is, of course, the actual genetic constitution of an individual, either with respect to all genes or a particular gene. And the phenotype is the outer observable manifestation. So here you see a bunch of common characteristics, if I could get this to work, a widow's peak, the ability to roll your tongue, etc. Really common, observable phenotypic variations that we see in the human population. And then over here you see an interesting example of an identical twin pair where one of them trained as a long-distance runner and the other one trained with weightlifting. And you see that even though they're genetically identical, they're phenotypically quite different from each other. So this is a really important distinction, genotype and phenotype. And one way to think about complex disease is that there's a lack of genotype-phenotype correlation. And this makes it difficult to study the genetic contribution. So the complexities are reduced penetrance, where some individuals who have a disease influencing genotype remain unaffected. And why is that? Well, it's that the genotype is really insufficient to cause the disease. And the implication there is that the effects of other genes and environmental factors are required in order to manifest the phenotype. So that's the gene-gene and gene-environment interaction that we're talking about. Another part of this lack of genotype-phenotype correlation is variable expressivity. So this is a situation where you have a single genotype associated with various different phenotypes in different individuals. And you can see immediately that reduced penetrance is actually an extreme example of that, because the phenotypic manifestations range from nothing all the way to perhaps a very severe phenotype, so variable expressivity. And then on the other hand, we have genetic and etiologic heterogeneity. So this is in a way the converse, where we have a single phenotype that's observed in individuals with a range of different genotypes. And so, you know, the phenotype can have multiple genetic and non-genetic causes in different individuals. So, and Dr. Leibel last week used the term phenocopy, which is an important concept. It's an individual who has the same phenotype, but who has a different genotype. So this genetic and etiologic heterogeneity is important at multiple levels. If you start with a disorder, most complex diseases have both genetic and non-genetic causes in different individuals. Within the genetic causes, you have different genetic loci, different genes that influence risk. And even within a single gene, you can have various different variants that influence risk. So here we have allelic heterogeneity. The different genes we refer to as locus heterogeneity, and the genetic and non-genetic causes we refer to as etiologic heterogeneity. So an example of the etiologic heterogeneity with the disorder that I work on, epilepsy, and particularly temporal lobe epilepsy, we know that there are environmental causes of temporal lobe epilepsy, severe traumatic brain injury or infections of the central nervous system, for example. But also there are autosomal dominant genes that produce temporal lobe epilepsy. So this is etiologic heterogeneity. Then we also have locus heterogeneity, which is very important in all complex disorders. And here I'm using the example of Alzheimer's disease, where we recognize 3 different genes on different chromosomes, all of which, when mutated, cause a relatively early onset and very penetrant Alzheimer's disease. So these families really look Mendelian. Individuals who inherit a mutation are very likely to develop Alzheimer's disease. And then there are other mechanisms. ApoE is a gene on chromosome 19 with 3 common alleles in the population, epsilon 2, 3, and 4. And individuals who have 1 or 2 copies of the epsilon 4 allele are at increased risk of developing Alzheimer's disease. So that's a very different type of mechanism. That's a situation where the allele is a risk factor. So there are lots of people who have an ApoE4 allele and don't develop Alzheimer's disease, and there are many people who don't have an ApoE4 allele who do develop Alzheimer's disease. It's a risk factor. And we know of many other genetic variants that also are associated with increased risk. So another example of variable expressivity is in epilepsy. We're learning that the alpha-1, the gene that encodes the alpha-1 subunit of the sodium channel, SCN1A, it seems to be involved in many, many different types of epilepsy. Practically every study design that you use turns up SCN1A as a risk factor. And it was first discovered in studies of an interesting disorder called genetic epilepsy with febrile seizures plus. And in this disorder, where you look at individuals within the same family, all of whom have the same mutation in SCN1A, you see a variety of different phenotypes, ranging from typical febrile seizures that go away after age 5 or so, to a more severe phenotype called febrile seizures plus, and ranging, a whole lot of different types of epilepsy syndromes in these families. So that when the disorder was first described, you saw really large families like this one, where the different colors indicate different phenotypes that were observed within the same family. When I first saw this, I thought, how do you know that they're all the same cause? But then the investigators actually found the mutations that were causing the disease in the family, and indeed all of the affected individuals had that mutation. So it's a classic case of variable expressivity. And another example of both locus heterogeneity and variable expressivity in epilepsy, with the GWAS plus phenotype, there are actually 4 different genes that have been found to raise risk for GWAS plus in different families. And each of them is also associated with other phenotypes as well. So we see a very complex relationship between genotype and phenotype here. So another extremely important concept is gene-environment interaction. And here what we mean is that genes and environmental exposures act together to raise risk for disease. And we can test whether this is present by doing studies where we ask the question, does the effect of the environmental exposure vary according to genotype, or does the effect of the genotype vary according to environmental exposure? There are a number of different models that you can think about in terms of how genes and environment might act together. And here are 5 of those models that are pretty fundamental. In the first one, the genotype doesn't actually cause the disease. It raises risk for a factor that is usually considered to be an environmental exposure. So if you think about obesity, for example, dietary fat and genetic susceptibility and obesity, an example of this would be an appetite-increasing genotype that actually increases the level of dietary fat and then indirectly influences risk for obesity. Another model, Model B, is where an environmental exposure causes the disease, and the genotype actually doesn't cause the disease. What it does is it exacerbates the effect of the exposure. So here we would think about dietary fat causing obesity and maybe a fat metabolism genotype being involved, so that people who only had the genotype but didn't have dietary fat would not be at increased risk. In the third model, you have a genotype that causes the disease, and the environmental exposure doesn't have a direct effect on risk. What it does is it exacerbates the effect of the genotype. So now, suppose you had an obesity genotype, but that obesity genotype would have a smaller effect in people who were not eating a lot of dietary fat than in people who were eating dietary fat. The fourth model is where the genotype and environmental exposure are both required, so people only have increased risk if they have both. And then the final model is probably the one, it's certainly the one that's the most complicated to think about and to evaluate, and it's probably the one that is most prevalent too, where the genotype has an effect, the environmental exposure has an effect, but when they occur together, their effects are greater than additive. So it's useful to think about these different models and to think about study designs for distinguishing among them. And really, we haven't learned very much about these, although we know that they're important. There hasn't been enough research in this area so far. One of the best examples, and one that's still controversial, is in psychiatry, where a study in 2003 by Caspi et al. And was published in Science and received quite a bit of attention and quite a bit of interest, because it was a study of polymorphism in the promoter region of the serotonin transporter gene and its effect on modulating the effect of stressful life events on depression. Now, there's a lot of research showing that stressful life events increase the risk of depression. But what this study showed, and it was a very well-designed cohort study, a prospective cohort study in a cohort that was followed for a long time in New Zealand, the Dunedin cohort. So what this study showed was that the effect of stressful life events on depression was not very strong in people who had the LL genotype. It was much stronger in people who had the SS genotype. And in fact, if you looked at people with no life events, no stressful life events, there wasn't really any difference in their risk of depressive symptoms depending on their genotype. But there was a big difference depending on the genotype in people who had a lot of stressful life events. So this is one of the reasons people were interested in this, is that they thought, well, if you take into account the effect of the environment when you do studies aimed at finding genes that influence risk, you might be in a much better position to actually find those genes, because the phenotypes will be different in people with different genotypes if they have a high level of the environmental exposure. As I said, this is controversial, but it is one of the best examples that we have of a gene-environment interaction effect. And it's been extended to other psychiatric outcomes, and a lot of other research has been done on this area. So, having introduced some of these complexities, now I want to talk about a pathway for studying complex diseases that we use in genetic epidemiology. So it begins with asking the question, is the disease familial? And of course, we do familial aggregation studies to answer that question. And I'm going to spend a bit of time on that type of study because it's really the most epidemiologic of the designs that we use in genetic epidemiology. The next question is, what causes the familial aggregation? So it's not necessarily genetic if it aggregates in families. You know, there are lots of things that aggregate in families that aren't genetic. Money is a good example. Not in my family, but in some families, money runs in the family. The tendency to go to medical school or law school runs in families. Ebola runs in families. So we need to ask the question, what causes the familial aggregation? And we use twin studies or adoption studies as, you know, rigorous designs to separate genes and environment. If we see that the familial aggregation is due to shared genes, well, we want to know what kinds of genetic effects are there. You know, is it all complex disease? Are there Mendelian effects? Are there chromosomal abnormalities, et cetera? And there is a method called segregation analysis that can be used to look at the pattern of incidence of disease in families and see whether it's consistent with these different models. And then finally, we want to know, well, how do the genetic effects cause the disease? And that's where we start doing gene identification studies and functional studies to try to identify the genes themselves and figure out what they're doing to raise risk. So returning to familial aggregation studies, as I said, I'm going to spend a little bit of time on this. They are studies that are designed to evaluate the extent to which a disease runs in families. And one of the things that's important about this is to estimate the recurrence risk, that is, the risk in relatives of affected individuals. For most complex diseases, or for most individuals who have a complex disease, we haven't yet identified the genes. And so we can't identify the gene and tell a relative of an affected person whether or not he has it and evaluate his risk that way. In general, we need to have empirical risk estimates that are based on well-designed epidemiologic studies. And those are what we need for genetic counseling in complex diseases. These are also important for defining the phenotypes that are likely to be most informative for molecular genetic studies. If you're going to design an expensive study involving collection of blood samples and maybe doing sequencing, you really want to be careful about defining the phenotype in those studies so that you get the most bang for your buck, so to speak. And these kinds of studies are also important for studying the relationships between family history and other risk factors for disease. So by phenotype definition, what I mean is which disease characteristics predict a greater genetic contribution. For many disorders, we find that the early onset variant of the disorder is more genetic than the later onset. We see that in a lot of complex disorders. But there can be other clinical characteristics that we can identify that help us to identify individuals who are more likely to have a genetic susceptibility, given the etiologic heterogeneity. And also, phenotype definition means answering the question, are there shared or distinct genetic influences on different clinically defined subsets of a disorder or on different related disorders? So here's what I mean by that. If you think about generalized versus focal epilepsy, for example, one model might be, and this is the model that probably most people working in the field have in mind, that there are different genes raising risk for each one. Alternatively, we might have shared genetic influences on different clinically defined subsets of a disorder. And this also applies to related disorders. So with epilepsy, there's comorbidity. We know individuals with epilepsy are more likely to have depression than other individuals. They're more likely to have migraine than other individuals. Individuals with depression are also more likely to have migraine. So this trio of disorders seems to go together. And we want to answer the question, are there shared or distinct genetic influences on these disorders? Well, we can answer that question by doing familial aggregation studies. And this was actually done before the genes that raise risk for breast cancer were discovered. There were familial aggregation studies that looked at the relationship between breast cancer, ovarian cancer, and endometrial cancer. And they showed very clearly that breast and ovarian cancer went together, but endometrial cancer seemed to be separate. So it's a very useful approach. So the most common study design is to sample cases and controls, and then to assess the disease occurrence in the relatives of each group. And in this sense, the cases and controls we refer to as probands. And the definition of a proband is the individual who is first sampled for the study, independently of his relatives, and who leads to ascertainment of the family. So what do I mean by that? Here are two families. You see in each case an individual who is a proband. Here's a case proband. Who's a control this is a control proband. And then we have their relatives. Their relatives may or may not be affected. So we have here, just as a simple example, 2 affected relatives in the cases family and 1 affected relative in the controls family. So what we're doing in a familial aggregation study is sampling the cases and the controls as probands, and then enumerating their family members and looking to see how many of their family members develop the disease. In general, the methods for data collection fall into 3 categories. First, a family history study. So this is a study where informants, generally the cases and the controls, provide information about their relatives. So just like you're asking them if they smoke, you ask them about their relatives. You know, how many relatives do you have? Is each one of them affected? And you can design a careful interview to try to ascertain the disease in the family members. And this is the most cost-effective approach, but the problem here is that for many disorders, there is a surprisingly high level of underreporting of disease in the family. And so this is low sensitivity. That is, many of the relatives who truly are affected are mistakenly reported to be unaffected. And there are also potential reporting biases with this approach that I'll talk about in a minute. At the other extreme, we have a family study where each relative is interviewed or examined directly. And you can imagine how labor-intensive that is. A study I did, we had 2,000 probands, and they had 10,000 relatives. So can you imagine going out and interviewing or examining each of those 10,000 people? Quite a lot of work. This is the way you maximize sensitivity and specificity of the information on the disease in the relatives, but it's really expensive, and it's also vulnerable to selection bias because only living relatives who agree to be participants in the study can be included. So that's really a limitation there. And so most studies are a hybrid between these 2 designs. A third design is a record linkage study. And here, information about both the disease and the family relationships can be obtained by reviewing linked, computerized medical records. So here you don't need to interview anyone. But there are limitations here because it can only be done where you have access to a linked medical record system that allows you to reconstruct the families from the data. And a couple of examples, the only one I know of in the US is the Rochester Epidemiology Project at the Mayo Clinic. And there are a lot of possibilities with the Scandinavian linked medical records. And another limitation here is that we can only do this with disorders that you're confident would receive a medical diagnosis and therefore show up in the medical records. So if you were trying to do it with migraine, forget about it. A lot of people with migraine are never diagnosed. Same thing with depression and many other psychiatric disorders. So you have to be secure that the disorder will be diagnosed. An example from the Rochester Epidemiology Project, I've laid out the steps here so you can have a better idea. imagine that you could identify individuals who have a disorder by screening the medical record linkage system using a set of diagnostic codes. And then you would select the cases that were born in the area and then use the birth records of those cases to identify their parents, and then go to the parents' records to identify all of their births, so that you would then have the siblings of the patients, and then review the medical records of the siblings to establish whether you had affected siblings. So I've done this, and I'll show you some data from that. I think it really, it turned out to be much more difficult than I thought it would be. It sounded really easy from the beginning, but it's an excellent way to study a disorder that is medically diagnosed. So what are some sources of bias in family history data? And by family history data, remember, I mean the indirect reports of who's affected in the family. Well, one of the most important ones is family information bias. So here, affected individuals may be more aware of their family history than unaffected individuals are. And this is really a problem. It's certainly the case for most disorders. For most disorders, women are more accurate historians than men. Timing is an issue, so recent events are more likely to be reported, or recent diagnoses are more likely to be reported. And relationship to the proband is an important issue here. So knowledge of the family history is clearly much better for close relatives than for distant relatives. Reporting of disease in previous generations is affected by secular changes in nomenclature, stigma, other issues. And some informants are better than others. For example, it's been documented in a study by Myrna Weissman that offspring are more accurate historians reporting on psychiatric disorders in their parents than parents are reporting on their offspring. And it makes sense, right? Because, everyone knows their parents are crazy, but, parents think their offspring are perfect. So to illustrate some of these problems, I'm going to tell you a bit about a study that I did of migraine in families of people with epilepsy. So these were about 2,000 people with epilepsy who were interviewed about their families, asking about severe headaches and migraine in each of their parents and siblings. And we asked two questions. Did the relative ever have severe headaches? Very general, broad question. And secondly, if the answer was yes, were they migraine headaches? A much narrower question. Then we also interviewed about 1,400 of the parents and siblings directly about their own headaches and the specific symptoms of their headaches, so that we were actually able to make a diagnosis of migraine, yes or no, in the relatives. And then we looked at the sensitivity of each of our interview questions for picking up the migraine diagnosis. And what you see here, remember, sensitivity is if a relative really is affected, what is the likelihood that the family history report was correct in calling him affected? So we found sensitivity was only about 56% for the very broad question, did your relative ever have severe headaches, when we're using as the outcome whether the relative had migraine. And then when we looked at whether if the relatives who didn't have migraine, and we looked at specificity, the proportion of those who were correctly reported not to have severe headaches, and we found specificity was about 83%. When we did the same thing using the more narrow question, did the relative have migraine, as you might expect, specificity improved. Lots of headaches aren't migraines, so specificity improved, but sensitivity dropped. And notice that it's very low, the sensitivity. We get a lot of underreporting here. Then we looked at a range of different factors that might predict sensitivity and specificity. And what you see here is that sensitivity is a bit higher for females than for males. And people who actually had headaches, the probands who had headaches, had higher sensitivity than those who didn't. And this is the family information bias that I mentioned. But equally importantly, the probands had lower specificity if they had headaches. So they were actually reporting headaches in their relatives if they weren't there. And this is really a problem, because you see a huge number of relatives who don't have the disorder. So a little difference in specificity can have a big impact on the findings. So they had higher sensitivity, but lower specificity. And people reporting on their parents had higher sensitivity than those reporting on their siblings. And that's probably because of the age at onset distribution of migraine. You know, if you're asked about migraine in your mother, well, you've grown up with your mother, you've seen her over the years disappearing into a dark room periodically, but your siblings may not have had onset until after you left home or after they left home, because migraine has onset, you know, in early adulthood or late adolescence in general. And here's another example of a study of Parkinson's disease where they evaluated the validity of the reported family history and found sensitivity was higher in the cases than in the controls. And just as I showed you before, a very small difference in specificity, but nonetheless, the cases also had lower specificity than did the controls. And then when they actually evaluated the level of familial aggregation, and this is the odds ratio for Parkinson's disease in the relatives of cases versus the relatives of the controls, they found that if they used the family history method, the disease appeared to be much more familial than if they used the family study method, where they actually went out and examined each person. So these are data from my familial aggregation study using the Rochester Epidemiology Project. And you can see that these are valuable because they give you an estimate of how the risk of developing epilepsy increases with age, here on the x-axis, in each class of relatives of the cases. And here we have the population level, so that we could calculate a standardized incidence ratio over here, showing about a 3-fold increased risk to each class of relative. And this is very important because we can't actually do genetic testing and tell people whether or not their relatives will develop epilepsy yet. Most people don't have a family history, and we don't know the genes. So it's about a 3-fold increase in risk, and the cumulative incidence is about 4% in the relative of an affected individual by age 40. Now, it matters what kind of epilepsy you're talking about. So if you're talking about idiopathic generalized epilepsy, the risk is much higher than if you're talking about focal epilepsy down here. But even in the very highest group, the risk in the relatives is less than 10%. So this is a very important piece of information to be used clinically, because you don't want to overestimate the familial risks. You need to have accurate estimates. And in this study, we also looked at the question about shared versus distinct genetic susceptibility. So here, when the proband had generalized epilepsy, there was quite a large increase in risk for generalized epilepsy. and a smaller increase in risk that was not quite significant for focal epilepsy in the relatives. And when the proband had focal epilepsy, there was actually no increase in risk for generalized epilepsy, but a 2 to 3-fold increase in risk for focal epilepsy. So, these data are informative for the shared versus distinct question. They do provide evidence for some distinct genetic influences on the two types of epilepsy, although this number suggests that there may also be some shared genetic influence. So now I'm going to shift gears and talk about strategies for gene discovery. And first, we need to think a little bit about why we want to actually discover the genes. So there are basically two ideas here. One is that we've said for years that we want to understand how the genes are influencing risk, so to obtain novel biological insights that will ultimately, we hope, learn to help us to develop better therapies, biomarkers, and maybe even prevent the disorder to begin with. But on the other hand, and this is why we're all here today, we also want to develop improved measures of individual disease processes. And this is where we're talking about personalized or precision medicine to develop diagnostic, prognostics, and optimized therapies. So there are 3 billion nucleotide bases in the human genome, about 20,000 genes, a much smaller number than we thought about 20 years ago. And 99.9% of these base sequences are the same across the population. So because we have 3 billion base pairs, that 0.1% implies that there are about 3 to 4 million base pairs that are different between any two people. So it's those differences that we think are related to disease susceptibility. Well, that are related to disease susceptibility. And there are a number of different types of variants that we can talk about: single nucleotide variants, structural variants. But the largest class of variants are the single nucleotide polymorphisms. This is the most common type of genome variation. We have about 1 in every 300 bases is a variant. And so there are about 10 million of them in the genome. And millions have been identified and put into databases now, so that they are an excellent tool for gene discovery. There are two major hypotheses that we talk about when we talk about genetic architecture. One of them is the common disease, common variant hypothesis, which says that common complex disorders are caused by common genetic variants, each of which has a tiny effect on risk. And then there's the common disease rare variant hypothesis that says that common complex disorders are caused by rare variants, sets of rare variants, each of which has a modest effect on risk. And which one of these you ascribe to has an enormous selection on the study designs that you choose. So what I mean by the common variant idea is that if each one of these lines represents a gene, or a sequence of DNA, you might have a situation where there's a variant which is always at the same position, and it's simply found more often in cases than in controls. On the other hand, you might have that same sequence of DNA, and you might have a situation where there are different variants at different positions in that stretch of DNA. So each one of these by itself is rare, but there are still more of them in cases than in the controls. If the situation on the left is what's happening, then you have a common allele with low allelic heterogeneity, and you can do allelic association studies to find those genes. If we have a situation like the one on the right, well, you can't do allelic association studies, which I'll talk a little bit more about in a minute. Instead, you need to do linkage analysis or genomic sequencing to find the genes that are involved in increasing risk. So, and here's a summary. We can do linkage analysis followed by sequencing. We can do association studies, either with candidate genes or genome-wide, or we can do next-generation sequencing, which is really what we're doing more of now. So with positional cloning, what I mean is you find families with multiple affected individuals. Then you carry out a statistical method called linkage analysis in order to localize a gene to a small chromosomal region. And then you sequence all the genes in that region in order to identify a mutation in one of them that's increasing risk. And the linkage analysis is simply asking what is the chromosomal location of a susceptibility gene. So you examine the inheritance of genetic markers, which are SNPs, in families with affected individuals, and then you test within each family whether the affected individuals have inherited a particular marker allele more often than expected by chance. And you test this using the LOD score. If you get a LOD score above 3, that's significant evidence for linkage. Now, this analysis is carried out within families. So these are data from the 1990 breast cancer linkage paper that eventually led to the discovery of BRCA1. And all of these families are showing some evidence for linkage to a genetic marker on chromosome 17q. But each family shows a different allele of that marker segregating with breast cancer. So in this family, it's the E allele. All of the affected individuals have E. In this family, it's the B allele. And in this family, it's the A allele. So each family has a different allele of the genetic marker segregating with the disease. That's what I mean by it's a within-family analysis. They're all showing linkage to the same marker, but a different allele is co-segregating with the disease in each family. In contrast, allelic association studies are asking, is the disease occurrence in the population associated with a particular genetic variant? And the classic example is autosomal Alzheimer's disease and the ApoE locus. That's an association. People who have Alzheimer's disease are more likely to have an E4 allele than are people without. That's not a within-family association, that's across families. So with linkage analysis, we're looking at families. With association analysis, we're looking at a bunch of unrelated cases and a bunch of unrelated controls, and we're asking the question, what is the frequency of each allele in each group? There is a huge industry now doing this genome-wide, starting really in the early 2000s. People started asking the question, can we find SNPs that are associated with disease that will help us to identify the genes that are actually causing the disease? So these studies survey the entire genome for common variants that are associated with disease, and they genotype up to 1 million SNPs in order to do that, and then test for association with each SNP one by one in very large case control studies. And so when you do this, you need to have a very stringent significance level, and the standard now is to use p less than 5 times 10 to the minus 8 as the level of significance. This takes into account the huge number of SNPs you're testing one by one, and also the lack of independence of SNPs that are tightly linked to each other in the genome. So what you see here is a Manhattan plot. This is how the data are arrayed from genome-wide association studies. And over here you see a skyscraper that is giving us evidence for association of Alzheimer's disease with the PCALM gene. I think it's on chromosome 11. And this is there have been much larger studies of Alzheimer's disease since this paper was published. This was 2009. Now there are studies involving even larger numbers of patients. And there's an accumulation of GWASs that have been done. So these are data from the NHGRI website that keep track of all this. And you see a huge explosion of the number of GWASs that have been done over time. By 2012, 1,350 of them. Now, in general, when you look at these studies, the odds ratios are quite small for the SNPs that have a significant association. So this is the odds ratio. This is the reported risk allele frequency, and most of the studies are showing odds ratios down or below 1.5, really. So the question has been raised, how important are these from a clinical point of view? You know, if you find a SNP that raises your risk 1.2-fold, how informative is that? Do you really want to know that? How important is that? And in fact, concern has been raised that when we evaluate the familial aggregation of a disorder and we have an idea about how heritable it is overall, what contribution are these SNPs making to that overall heritability? And for most disorders, the heritability is the contribution to the heritability of the identified SNPs is quite low. There are some exceptions. The macular degeneration example is really the best example of one with a high contribution, but most disorders, it's rather low. So that is why really we are now turning to sequencing in order to find with greater power rare variants that are contributing to disease, because these rare variants probably have a greater effect on susceptibility for individual patients. And I'm going to stop there now with the hope for sequencing studies in the future. Thank you very much.